Hello everyone, welcome to WinPoint online classes. In this session, we are going to discuss the methods of asexual reproduction. Let's begin. The learning objective for this session will be methods of asexual reproduction, in which we will be discussing about two methods. One, the fission and its two types, binary and multiple fission, and budding, its two types, endogenous and exogenous budding. Asexual reproduction is a type of reproduction in which single parent is involved. And we have already seen that the offsprings so formed are the same as that of parent. Let's discuss the methods of asexual reproduction. Fission. Fission is a type of asexual reproduction in which the parent body splits or breaks into two or many daughter cells. Based on the number of daughter cells, the fission is being categorized into two categories. The first, binary fission, and the second, multiple fission. Binary fission. Binary means two and fission means splitting. So the definition is, binary fission is the type of asexual reproduction in which the parent cell will divide into two daughter cells. Now let's discuss the features of binary fission. First, we have already discussed that the one parent cell will split into two daughter cells. Now, what is the process going on? First, the nucleus divides. The nucleus which is present inside the cell will split into two. This process is called as karyokinesis. And later on, the cytoplasm divides into two daughter cells. This process, when the cytoplasm divides, is called as cytokinesis. So we have learned two terms, karyokinesis and cytokinesis. Karyokinesis means that the nucleus divides, and cytokinesis means the division of cytoplasm. So in binary fission, the parent cell, first inside the parent cell, the nucleus will divide, and later on the cytoplasm divides and forms two daughter cells. All organisms undergoing binary fission are immortal. Immortal means that they don't have a natural death, but accidental death is possible. Example, it occurs in amoeba, bacteria, and even some algae. Now let's discuss the example of binary fission in amoeba. You can see the structure of amoeba. It has an irregular body shape. Now when it undergoes binary fission, you can see that first we have learned that nucleus divides, later on cytoplasm divides. So the parent cell, you can see that in the next stage it gets mature, and you can see the nucleus is dividing and nucleus is split into two. Later on you can see that the cytoplasm had divided and it forms two cells which are still attached, and later on it splits into two daughter cells. So this is binary fission. Inside the parent cell, the nucleus will divide, and then the cytoplasm divides, and later it forms two daughter cells. Let's move on to the second example. You can see the structure of bacteria. In this structure, you can see a stage where the bacteria parent cell is splitting. Now let's understand the binary fission in bacteria by a simple example. You can see a structure of bacteria. It gets matures and it gets developed. Later on, you can see that parent bacteria cell will split into two daughter cells. Now these two daughter cells are capable of becoming a parent and splitting into another two daughter cells, making four daughter cells. So on, the reproduction continues. Now let's discuss the second type of fission, that is multiple fission. Multiple means many and fission means breaking or splitting. So the parent cell will split into many daughter cells. So let's see the definition. Multiple fission is the type of asexual reproduction in which the parent cell divides into many daughter cells. We have studied previously that binary fission is a fission in which the parent cell will divide into two daughter cells. Now in multiple fission, the parent cell will divide into many daughter cells. Now let's see the features of it. Is the same feature what we have discussed. One parent cell splits into many daughter cells. Now, what is the process which is going on? First, the nucleus will divide and forms many nuclei. But in the case of binary fission, it will form two nuclei. But in multiple fission, it will form many nuclei. Then, it is surrounded by the cytoplasm and a membrane. Later on, the parent cell ruptures and all the daughter cells so formed will be moving out. This is the general process of multiple fission. It occurs in Plasmodium, which is a malaria parasite. Now let's discuss the example. Multiple fission in Plasmodium. You can see the structure of a Plasmodium. It is having one nucleus, cytoplasm. It is surrounded by cytoplasm and a membrane. Later you can see that a many nucleus is formed along with the cytoplasm and a membrane inside the body. It forms a central mass. Later on you can see that it ruptures and the inner cell mass, that is cell, all will be moving out. This is how the multiple fission will take place. Let's repeat. A parent cell forming daughter cells inside the outer covering, and then the outer covering ruptures and the cell moves out. This was about fission. Now let's discuss the second method of asexual reproduction, that is budding. Bud is generally an outgrowth or a projection which is formed on the parent body or inside the parent body. So what is the general process? A bud is formed. Later on, the bud develops and it gets detached from the parent. So let's see the definition of it. In this method, a bud-like projection or an outgrowth is formed on the body of the organism. The bud develops and then separates from the parent to form an independent individual. Budding. Budding is of two types based on whether the bud is formed inside the body of an organism or whether it is formed outside the body of organism. Based on that, the budding is of two kinds. First, exogenous and second, endogenous type. Let's discuss exogenous type. Exogenous budding is the budding in which the bud is formed on the outer side of the parent body. Example, yeast. Let's see the structure of yeast. A yeast cell, it grows, then an outgrowth is formed. You can see an outgrowth, and later on you can see that a new bud is formed. The bud is getting matured and then it gets detached from the parent body. This is the process of budding in yeast. Yeast cell, which is a parent cell. On the parent cell, a developing bud is there. The developing bud grows and forms a new bud, and when the bud gets mature, it gets detached from the parent and forms a daughter cell. Let's discuss another example, budding in Hydra. You can see the structure of Hydra, in which the bud formation is taking place in the outer surface of the body. Later on, the bud gets mature and gets detached from the parent to form a new individual. Now let's discuss the second type of budding, that is endogenous budding. We have already discussed about exogenous budding, in which the bud formation will take place at the outer side of the parent body. Now in endogenous budding means that the bud which is formed will be formed inside the parent body. So the bud formation is inside the body of an organism. Example is Spongilla, which is a freshwater sponge. Endogenous buds are known as gemmules. Now let's see the structure of Spongilla, that is sponge. Now let's discuss the structure of sponge. It is having outer porous body. Then you can see that inner body wall is there, which is having gemmules. These are called bud. Gemmules are also known as bud. And you can see an opening through which these gemmules will move out to the water surface, and later on it will form a new sponge. Now let's see the structure of gemmule. body mass which is consist of many cells. These cells are called archaeocytes. So archaeocytes are the cells which are present inside the gemmules. You can see the outer opening which is called micropyle, through which these will be attached to the substratum and the cells will be moving out. You can see a spicules, there is exoskeletons or endoskeletons which are called spicules, which will be helping or which will be protecting the cell. Whereas you can see the gelatinous covering which is present, which will be preventing it from drying. In the next session, we will be learning about spore formation, fragmentation and plasmatomy. Thanks for watching.